Hello, everybody. Welcome to CaveNet and this says webinar series. We're glad everybody's able to arrive. We will have our webinar tonight called Uncovering Secrets of Silk Used by Cave Spiders and Glowworms. This is very interesting. I've already seen some of the slides, but our presenter tonight will be Dr. Dakota Perowski. I talked to him a little. He's got an interesting background. I'm from Akron, Ohio, but now he lives in Taiwan. It's been there for a while. But I'm going to go ahead and talk about him. But first, I want to thank our sponsors, NSS. Without them, of course, we wouldn't have a webinar. And I'm Edward Zuckerford. And I'm Debbie Spoons, the chairperson for the webinars. And I'm going to talk about Dakota. There we go. Sorry, technical. Okay, now, Dakota is a postdoctoral research fellow at the Department of Life Science in Donghai University of Taichung in Taiwan. He received his PhD in biological materials research from Donghai University in June of 2019. I received a Bachelor of Science in Biology from the University of Akron in Akron, Ohio in 2013. And I asked him how he got from Akron to Taiwan, and he was giving me his background. It's very interesting. Now, Dakota is a scientist trained at the intersection of the fields of biology and physical mechanics, and he specializes in the study of the amazing properties, features, and characteristics of naturally occurring fibers, including spiders and insect silks. And all of us cavers know about cave spiders, and we've all run into these amazing structures. If you look at them closely, they're very intricate and detailed. Dakota conducted research studies during 3 trips to the karst cave systems of Tasmania, Australia. Though he does not have immediate plans to conduct further studies in cave systems, Dakota is always open for an opportunity to get wet, cold, dirty, and collaborate with cave dwellers. So he's just, sounds like our type of person. So anyway, what I'm going to do now is go ahead and turn this webinar over to Dakota, because he's got some beautiful pictures and some amazing information for us. If you have any technical difficulties, use your chat button, and remember, everything is recorded and stored on the NSS website at caves.org. All right, Dakota, I'm going to go ahead and turn this over to you, and you can educate us.
Okay, thank you so much for the introduction, Debbie. Once again, I'm Dr. Dakota Perowski. I'm here to share with you today studies from my PhD thesis itself. I'm going to be sharing 3 different studies, all involving the wonderful spiders and glowworms of Tasmanian caves. And hopefully you'll find them as interesting as I did, and you will maybe take a second look at spiders next time you're walking through a web. Maybe they'll change your mind a little bit on how scary they may or may not actually be. So today I'm going to start by talking about the cave environment and the animals that live there. They're very unique in many ways, and hopefully this audience knows that, but for those that don't, I'll provide a very brief introduction. Then I'll go into the amazing properties that we know of for insect and spider silks, and there are probably a few that will surprise you. And then we'll get into the studies that I actually conducted, the primary research that I've uncovered, along with the team that I worked with, some very wonderful people, both from the materials and physical sciences world to actual cave people, along with the videographers and film directors that helped me both find funding and provide photos and help me become involved in their movie. And that will be 2 different topics. There's one study about life growing up in the cave, and then 2 other studies about how spiders and glowworms are able to produce adhesion and sticky silk in the cave. And then finally, I will close with a few remarks about how caves can be a new frontier, and not just for the current understanding we have in terms of understanding zoology, biodiversity, conservation, but potentially for biomaterials and for a new class of people that may be interested in the wonderful things that happen within caves. So we'll start with talking about the cave environment. So for anybody that's been into a cave in the past, one thing that will be immediately obvious is the low light settings that occur. Obviously, except near the entrance, most of the cave is not very well illuminated. This has obvious consequences for any of the animals or any life that inhabits these caves, as this leads to very low food production, as bacteria and plant life often requires sunlight for photosynthesis, and this is the bottom chain of the food web. A lack of this primary production leads to a lack of higher orders of animals that can live there. Another consequence that makes life difficult is the constant high humidity in wet caves. So any caves with streams in them. This often makes it very difficult for things like arthropods to live in them, as they have evolved adaptations for conservation of water, which would cause them to effectively drown at such high humidities. And this makes it very harsh for most animals to live in caves permanently or otherwise. And as I said, most of the light and animals that can inhabit caves do exist toward the entrance zone of caves. This is where you'll find temporary inhabitants and some green vegetation. And as you go deeper and deeper the cave, there's less and less light. Temperature becomes more constant and stabilized, but less and less life can inhabit these areas. However, for the animals that can live in caves, they're subdivided into three different categories. The first being the troglazines, and these animals are temporary and can spend their entire life outside of the cave, but do choose to spend some period of time inside of the cave. This includes bats, extinct animals such as the cave bear, and potentially not extinct, though popular media might say otherwise, the caveman. Now, troglophiles are animals that can live both, that do need to spend, they do prefer to spend time in the cave, but they can spend some time outside of the cave given proper conditions, such as humidity in the case of glowworms, and then cave spiders. They can spend some time outside of the cave, but they can also spend their entire life cycle within a cave as well. And the troglobites, which are the most unusual and interesting animals, are completely dependent upon the caves for their life cycle, and they do require cave conditions constantly. And these are the most interesting ones because these are the ones that exhibit very weird adaptations. Some of those include a loss of pigmentation, as we can see from these cave salamanders and cave fish, and as we'll see, glowworms. A lot of these animals exhibit a reduction in eye organs or simply a loss of eyesight. Again, as the case goes with the cave fish and in some cases cave salamanders. Elongation of limbs, reduction of metabolic rates, increased lifespan are also very common adaptations exhibited by animals that live in caves. And this is largely due to the lack of light, as well as the lack of food. They need to adapt to survive under these harsh conditions. Production of light is also seen by bioluminescence in these glowworms. And we are looking at for the existence of we're currently inhabiting, currently looking at how these cave animals are still producing interesting adaptations. Now, caves have long been described, or at least within the past 50 years, as natural laboratories. These are stable and definable climates, and that they don't day to day or month to month, year to year, we don't see much change in the environmental conditions. And this allows for easier study of these kinds of environments, because they're make the food web simplified, where we can then understand where nutrients are flowing, where they're coming from, and disturbances are quite minimal, so that except for flooding events, generally speaking, there are few instances of large-scale environmental changes, and this allows for a much easier study of the animals that live there and their interconnection. This allows for us to understand physiology, ethology, or animal behavior better, ecology, biogeography, and in the case that I'll be presenting today, for biological materials. Because the systems are so simplified, we are able to better understand how and where these adaptations might be coming from. And in order to understand these biological materials, we do have these candidates that I've studied, and other people as well, spiders and glowworms. And I'll give you a much better introduction to these animals a little bit later in the talk. But one thing you should understand is that these are top predators, because these animals live in caves that do not have bats. These spiders and glowworms are at the top of the food chain and are able to consume insects. And they build silken traps. They use silk to produce webs and to produce what looks like fishing lines, or as I like to call it, a silk curtain. And they are dependent upon their silk. They cannot use any other means to catch prey. They need these sticky threads and the structure of their trap to be able to mechanically arrest insect prey. So with that, let's have a brief introduction to the topic of silks and the amazing things that they can do. In most insects and spiders, which collectively are known as arthropods, silk is a building material. They use it largely to build nests and cocoons for both metamorphosis, as well as producing a protective barrier from the outside world for the eggs that they lay. But in many cases as well, they produce webs and traps, such as orb weaving spiders, as you can see here, eating a fly, as well as the glowworm fishing lines. Now, other lesser known attributes of silk are also for safety lines. As you can see, many salticid jumping spiders use their silk lines to stabilize their jumps and use it as direction for where they're leaping to. They also are used to disperse pheromones to allow males to find the females as they're wandering around their environment. And then obviously they use these silks to catch prey, particularly those that build webs and traps. And as you can see from our nice little GIF here, from the cartoon of Adventures of our Adventure Time, Finn the human is jumping into webs. Whenever I present these to students, they always are able to recognize this and really enjoy seeing it. But these webs are very, very strong and can catch many things. There's many examples of spiders catching things that are much bigger than you'd expect, such as large insects, birds, bats, snakes, reptiles. And the glowworms do something similar, catching whatever they can that flies into the web. They're not these animals are not particularly picky. Now, in the case of spider webs, they use, well, spiders can produce more than just orb webs, the traditional vertical sheet that you may have walked into at some point. They can produce horizontal webs, triangular webs, vertically elongated webs, cobwebs, three-dimensional webs, sheet webs, funnel webs. There's all kinds of different webs. And for most of these spiders, in fact, just about all of them, those that use webs to catch prey, this is their only source of prey capture. They don't, except in very rare instances, they don't tend to wander out of their webs and actively seek out prey. They stay within their webs, and the only changes they have is that they may find ways to they may relocate their web and find better places to hunt. However, once they're in their web and once they've produced it, they're totally dependent upon it. And in most cases, and the two most prominent silk types that are important for these webs are their major ampullate silk, the dragline threads. These are the primary structural support threads, and I will be spending time talking about this silk thread in the first study. And then there's sticky capture threads, of which there are two primary kinds in different species. Each one uses one, whether it be a glue-based adhesive or a dry nanofibril adhesive, and I'll spend time talking about both later as well. But these are the two primary kinds of silk that are important to spiders, and the two primary types of silk I'll be focusing on in this presentation. So, as I said before, spiders are capable of catching very large things in some species, such as these Nephila, which here in Taiwan we have quite a few of them, though I'm yet to see any spiders catching anything really big, but there are reports of this occurring. The primary thing that people know spider silk for, and what seems to be grabbing the attention of people in popular culture, are the fairly amazing mechanical properties of silk. So spider silk combines a organized hierarchy at the molecular level that provides both lightweight properties as well as these this combination of strength and extensibility. If we look at our graph here, on the x-axis we see strain. This is just a percentage of original length, how far something can stretch until it breaks. So obviously, 0.20 would be 20%. Stress is a normalized value of strength. It's force over cross-sectional area. So this is the amount of energy or the amount of force that can be applied to something before it ruptures in this case. So the end of the lines mean the point at which these fibers break. And if we look closely, we can see that these three names here, Arrhenius, Latrodectes, and Nephila, these are all spiders. This is spider dragline silk. And you can see it has this combination both of strength and extensibility, where it allows it to outperform Kevlar, in which Kevlar is very strong but not very stretchy, up to possibly 2% of its extensibility. And then nylon, which is stretchier but not very strong. Stretchier in the case of stretchier than Kevlar. These two are Bombyx mori. These are silkworm silks, and as you can see, not as strong but also stretchy. And spider silk, given these properties, is the toughest known biological material to date, and we're constantly trying to replicate these properties. However, there are several other properties about silk that are interesting to scientists and biologists such as myself. Now, as I said before, what makes spider silk so interesting, or how it produces these mechanical properties, are through its hierarchical molecular structure. There's a combination of nanocrystals, as you can see from this cartoon, these sort of brick-looking structures. And these are beta-pleated sheets, so imagine protein sheets that are folded on top of each other. This is what gives silk its strength. The amorphous structures, which are sort of interconnected, semi-crystalline, kind of gooey almost, the more fluid-like structures, are composed of alpha helixes and beta turns. This is what gives silk its extensibility. So if you can imagine, you have a bunch of cords and bricks that are interconnected, and when you stretch them, it allows for a little bit of give, but most of the force gets dispersed to the strong crystals. So this is what gives silk its toughness attributes, as it's strong and stretchy. But it's also, in the case of capture threads, it's also very sticky. Now these are the spirals in the orb web and the fishing lines that I'll talk about later in the glowworms have similar properties, but these capture threads are coated in an adhesive. And in the case of viscid silk, like in many orb webs you see, it's a glue. It's an aqueous glue that coats along the thread, and they form into these droplets due to Rayleigh instability. But another type of silk is cribballate silk, and these are formed as they're tiny nanofibrils that are at the diameter scale of 10 to 100 nanometers. They're very, very fine, very, very small. I'll talk about these in greater detail later, but they both are sticky, and they can produce some very beautiful photos. As you can see here, they're very responsive to the environment. Changes in humidity will change their behavior. As you can see here, that's quite a cool difference. In dry conditions, in some species, the droplets stick directly to substrates and don't necessarily deform very much. At increasing humidity, they become more and more liquefied and then deform at greater levels. It's really something quite remarkable, and there are many studies recently that have been done that exemplify this. Now, spider silk, such as, again, dragline silk, have many cool properties that we're only recently starting to uncover. One of which is the high resonant frequency of spider silk. And this is interesting for engineers and material scientists, but also for biologists studying zoology. If we look at this left graph, because I know it's a lot of information to present on just one slide, you can see spider silk here along with silkworm silk that have this longitudinal wave speed near 3,000 meters per second. Longitudinal wave speed is the speed at which vibrations travel through a material from molecule to molecule. They're compression waves. And this is dependent upon the resonant frequency. So you can see that it's very, very high, quite similar to copper wire or a metal, and much higher than nylon. And the reason this is significant is because many materials that are used in construction and building, such as tubing, needs a high resonant frequency, because low resonant frequencies, which is where most ambient noise it comes from, can cause destructive resonance, can cause resonance that destroys the material when you use it. So having very high resonant frequencies is quite useful if we can capture those properties. Other interesting properties from dragline silk are the antibacterial properties exhibited and recently shown by our laboratory, where these are, if we look at this graph, I know it's a little bit complicated, but the basic thing to understand is that we used silk from 3 different spider species, and these are species that all exhibit, they live in different habitats, they produce different kinds of webs, and they potentially eat different kinds of prey. And when we expose them to different bacteria, we use 4 different bacteria in a control, and then we use different experimental groups where LB and NFG are just positive and negative control. So LB, you expect to see growth because this is full nutrients. And PBS is the negative control, which has basically no nutrients. And this is the negative control. NFG is full nutrients without nitrogen, which is what we expect spiders to be using if they were using spider silk as a protein source, which is, again, or amino acid source, which is full of nitrogen, we can see that in almost all cases, bacteria are unable to use the protein of spider silk and do not grow. All of these response variables is the growth rate of bacteria, and we find that in 3 species, very little growth is observed, and so this is evidence that spiders have antibacterial properties. They're not necessarily killing bacteria, but they're preventing it from growing along their silk. And we're following this up with research, that project that we've just started, but this is all really cool stuff that, again, is very applicable if you look at, if you consider medical uses and medicinal uses of silk. But anyways, going back to how silk might cope in caves, in cave environments specifically, we want to know, as I had alluded to at the very beginning, how does spider silk, how is it changing over the lifetime of spiders? Ooh, sorry about that. How do the long lifespans of spiders in caves, how does that affect their physical properties? And then how are their adaptations of silk to the damp cave environment? Do they show preference, and do they have interesting properties that are influenced by the cave? Both of these are questions that we were very interested in and continue to be interested in. So we'll address that first question in one of the studies that I completed a few years back. Growing up in the cave, this study was conducted using this beautiful spider, which I'll spend a little time talking about in a few slides. But this work was published and got on the cover of the Journal of Zoology. So if you're interested, I can provide links and information to this paper if you have further interest in reading it. So, as I had said before, spiders are not only dependent upon their webs in adulthood, but they are dependent upon them throughout their entire life. For prey capture, for food consumption, just when they're waiting to catch things and resting, keeping their metabolic rates low because they aren't actively looking for prey. They're somewhat dependent upon their environment. And they do use these webs for mating as well. This is sort of their primary sensory system of spiders because their eyesight tends to be very poor, mostly in spiders that use webs. So these webs need to be functioning at high levels. As I had said before, the dragline threads and sticky capture threads are very important to these spiders. As they need them for survival, they must perform at a high level. This allows spiders to dissipate energy of impacting prey or other objects that might fly into their webs, to detect vibration of the prey or intruders, and support their own body weight. And like I said, the capture threads allow them to retain prey, but also in some cases collect water for them to drink. Now, there are many examples of very interesting changes across lifespan of spiders. In many cases, these webs start out looking like orb webs in that they are circular, but then transition to more refined versions of the web. In some cases, triangular or elongated, or more vertically asymmetric. And so there are many documented cases of this, but few cases of changes in the silk itself. We wanted to know, do the primary building blocks of these webs change in their properties? We know that the webs sometimes do in certain species, but do the silk properties change? This would be very important in understanding the ontogenetic effects, so the developmental effects of silk, and whether or not there might be any sort of engineering practicality as we could study the changes that might be occurring and understanding how that might be applied. However, there's just very few model species right now because there's a lack of research and therefore a lack of identification of species where we see changes in properties. Previous studies, and as I said, there are only a few, have not identified many shifts. We can see here on the x-axis, these are all increases in body mass, or body weight, and we can see that this line is pretty much flat. So we don't see shifts in properties across stiffness, strength, or extensibility. However, just not enough studies have necessarily been done. So enter our spider, Hickmania troglodytes, the Tasmanian cave spider. I've been building this spider up over the past period of time to talk to you about them. They are quite large web building spiders. So these spiders, we don't necessarily see any shifts in their web structure itself, other than it just getting larger. But in my time spent in Tasmanian caves, I've seen webs that are as big as mattress covers. These things can be, on average, 1 meter in diameter, but I've seen ones that are much larger than that, up to 2 meters in length and 1 meter in width. You could, this would fully capture a person if you were to use the whole thing. They use major ampulate silk, like I said, that dragline silk line, and these are used to be interlaced within the catching area, but they're primarily used as support and energy dissipation. So these are the primary silk threads that are performing work when when insects do fall within the web. And what's unique about this in the cave environment is that multiple instars, meaning multiple sizes of spider, from very young spiders to very old spiders, all cohabitate the webs, or the caves in these cases, and this allows us to take a snapshot and observe how spiders in this species might all be trying to catch similar prey that exist within these caves. Again, they may be selective, but they may be trying to catch similar things because of the lack of food in these caves. So in order to characterize spiders, we visited Mystery Creek in Bradley Chesterman Caves in southwest Tasmania. We measured the body sizes of spiders. We looked across their carapace width and their body length. These are standard measurements for measuring spiders. And we collected sections of web. This was probably the most fun part of the study, getting quite messy and squeezing into little crevices and corners of caves that are quite hard to access, but and often result in being quite cold for a long period of time. The average temperature of these caves is about 9 degrees Celsius, and these spiders build in very weird places that are quite difficult to get to, but you might be standing there trying to collect these webs with a slow, solitary drip of water right into your face while you're slowly sliding away in the mud. We use these cardboard frames with sticky tape and use scissors to cut away the frames, or the parts of the web. So we tried to be as minimally provide as minimal disturbance to the spider as possible, as they've become a bit of a symbol of caving in Tasmania. Once we collected the web sections, we then collected individual pieces of silk. As you can see from our photo here, that we would take light microscopy measurements to get diameters. This is to normalize our force measurements because we're looking across body size, so that obviously fibers from very small spiders would be, from the absolute force measurement, be weaker, but if you normalize them, it gives us a better understanding of the material itself. Once we have those diameters, we can then run our tests. As I showed you before, the goal is to measure tensile properties. And these are very typical stress strain curves, kind of like I talked about a little bit earlier, but we also can measure stiffness, which is this initial slope of the stress strain curve. Where the slope changes is the yield point. And again, stress and strain, just to reiterate, this is the y-axis is strength. It's indicated by where it breaks on this y-axis, and then how far it stretches is also the extensibility. It's a measure of stretchiness. So we use a very specialized machine called the Nanobionics. We measure it by mounting them between a clamp and a very sensitive force plate, and we stretch the fiber until breaking, and at the point of fracture is what we measure as our ultimate stress and strain. These values allow us to calculate all of these kinds of variables, from the immediate measurements of extension and load converted to stress and strain, then calculating the modulus from the slope and toughness as the area underneath this curve, which is the overall work completed by the fiber, the energy absorbed by the fiber. So when we measure the diameter, obviously diameter increases, as you can see by this is a measure of spider size. This is the measure, this is a measure of spider size and carapace length. This is just to be sure that both of those measures could be used in our study, but with diameter, as you can see, it increases, and it increased insignificant from isometry, which just means that diameter size increases at the same rate as body length, body size measurements. So we don't necessarily see anything particularly interesting in terms of the diameter growing faster or slower than the rest of the body. And when we compare across spider size, then we do see changes in these mechanical properties. So from a qualitative perspective, by just looking at exemplar data, we see that the small individuals produce silk that is, you can see, clearly different from those of larger and larger spiders. Not only do we see that the breaking stress increases, and that's exemplified statistically across body size, both in our measure of body length and in carapace width, but also the area underneath this curve, which is toughness, which is this work performed. Qualitatively, we can see that the behavior is also much different. We can see that in the largest spiders, these curves are very well developed and clean looking. As the spiders get smaller and smaller, they seem to be a little more erratic, which may mean that they're slightly underdeveloped. That we need to spend more time demonstrating, but we can clearly see that there is a shift, sort of a difference in these mechanical properties. And this has fitness implications for this species, but also it may have evolutionary implications. From the direct result, this may mean that spiders, as they grow, become better capable of catching more prey, as their webs may be better able to support more things in the web, potentially bigger things in the web. They may be more resistant to damage, either from captured prey or from items falling into the web, such as pieces of the cave, like rocks and stones. And this also may allow for better silk economy, meaning that spiders can use less silk in their webs compared to when they're young, and maybe at a lower energy cost. And also, finally, as the spiders grow larger, it may better allow them to support their own body weight and allow them to stay within the webs. We haven't directly tested the exact consequences, but these are things that we could look forward to testing in the future. And there are plenty of reasons to think that and be curious as to why these shifts occur, because they may be down to the composition, the chemical makeup of the silk may be changing. This may be due to changes in physiology of the glands that are used to produce them, or the physical characteristics, the structure of those glands in some ways that may be different. Identifying those changes, now that we know that there are these shifts, can lead to a better understanding both of silk properties in general, as we still know very little about how silk changes through spider development, but also can lead to a range of understanding a range of strategies that may occur, and also ways in which we might better develop artificial silk. This may inform us of how to build better silk spinning apparatuses, as well as providing better dopes or starting materials to produce fibers from, as we're still getting closer to producing artificial silk that replicates the properties of natural silk. We still just don't know enough. So now we're going to get into the largest section of the talk, which includes two studies, one that will get back to the cave spider and one that will finally focus on the glowworms and their quite magnificent sort of beauty and use of silk that is really quite unique to any other kind of silk that we know. These spiders are really these glowworms are really quite a spectacle to see. If you ever have the chance to go to Tasmania or New Zealand, to go into the caves that they inhabit. They also exist in the southern part of Victoria in Australia. These insects that when you turn your lights off in the deeper parts of the caves, it looks like stars in the sky, and it's really quite a majestic thing to see. The study that we conducted on these spiders was also published 2 years ago. I would recommend to check it out if you are further interested in the work that we did, and going to see more pictures. But these glowworms are called Arachnocampa, which is Latin for spider bug. These insects are the larval stage of the fungus gnat, so they metamorphose and become these kind of clunky, awkward flies. They are dipterans, and they're only found in dark, wet habitats like the caves, but they can also exist in some of the surrounding wet temperate rainforest. They use their bioluminescent lights as lures to attract wandering insects, often insects that are lost from the forest or might be inhabiting the cave at some point, or even they cannibalize the adults in some instances. And the silk is used to catch those wandering prey. So they get stuck on these sticky droplets, and like a fishing line, once they're stuck, they struggle and the thread stretches, but the glowworm goes over to and finds the thread that they're stuck to and starts consuming the silk line up until that prey, and then they consume the prey. The larvae themselves are kind of ugly looking and transparent, and to the point that you can see right through them and see all their inner organs. But from far away, when you look at the silk and when you look at the lights they produce, they're really quite beautiful. And our question going into this was, is the silk dependent upon the cave conditions? We know that these insects, these glowworms, are very responsive to dry air. They will be quickly dried out and die if they're exposed to anything less than cave conditions. They don't survive for very long. And in these sort of anecdotal reports, silk was also observed to become very brittle from people touching and pulling on the silk. However, very little empirical evidence has been provided. We wanted to then know, are they dependent upon cave humidity and cave conditions? Is not only their bodies, but also their ability to forage and catch prey dependent upon these environments? So we set out to understand how water affects these properties, and that in wet conditions we would see improved in the sense that they might be better able to absorb and dissipate mechanical energy, as well as be more adhesive for insect prey. Again, we went back to Southwest Tasmania along similar trips as when we were collecting spider silk from Hickmania spiders. And we would collect these silks onto these little cards directly front that were hanging. This was, really fun and easy because the silk just, you don't even need any glue. They just stick directly to the cards, and you can walk out, just cut the line. They are quite responsive to wind though, so breathing on these things will cause them to all stick together, but it's really quite a fun thing getting to go into these caves. And what we first did was measure adhesive properties as well as mechanical properties. And what we would do is take these threads, we'd cut away part of the card and press them down into a sensitive force plate. The same force plate we'd use for tensile testing. We'd press them down into the plate and then pull up on the force plate and measure the amount of force required to detach the silk. So it would be attached across these force plates, and that would make up about a 2 millimeter segment of these 10 millimeter thread segments. And as we pull up and pull off on the force plate, we would then know how far the thread is extending and how much work is being performed, how much energy is being absorbed during adhesion. And we would also then, using separate threads, measure tensile properties using methods that are the same as I described before. And we would also be determining diameter size. And this is a thread that's been pressed onto a glass slide, so we can measure the diameter directly. So we measured samples in 2 different environments at very high humidity, above 90% relative humidity, and at dry conditions, 30% relative humidity. And we gave samples at least a minute to acclimatize, and these were tested inside of environmental chambers where we could adequately control the humidity using evaporated air. Evaporated water in the air. So what we found when tensile tested at different conditions is that the mechanical behavior is very different. Dry silk becomes very stiff and not very stretchy, much like the silks, much like what I showed you with Kevlar, except it's not nearly as strong. However, when wetted, not only do they become much stretchier, they don't lose very much of their strength, and they become very compliant, meaning that we can stretch them and then return them back to their original starting position without much loss in properties, without overstretching them. If we were to stretch up to 20–30%, we could return them back to their original starting point and be able to stretch them again. Similar to how we might expect them to perform in nature, where we would potentially need to stretch and unstretch a struggling prey that would be caught in the webs. Now, as you can see, we don't see significant differences in stress, which is important because if they were less strong in wet conditions, this might be a problem, but as we can see, it's not. However, we can see very clear differences in every other property. So extensibility, obviously the dry threads do not stretch much at all. The dry threads are very stiff and brittle. They're not able to dissipate much kinetic energy, and they become clearly very stiff. And the wet fibers, these values are a measure of stiffness, and the wet threads become very compliant. They become very stretchy, and that high measure here would mean that they resist deformation very quickly. So imagine the difference between sort of ceramic clay as being high Young's modulus, whereas something wet, the wet silk of glowworms, will be something similar to a rubber band. So this is very similar to what we see in other silks from other types of animals. In insects, when wetted, silk strain, and this should actually say increase, but extensibility increases in wet conditions in both these lace wings that produce these stretchy egg stalks and caddisflies that build these casting nets underwater to catch prey, and spider silks as well, that when they're wet, we see an increase in extensibility as well as a increase in compliance. So water has these same effects, that when they're dry, we see reduction in extensibility and stiffening, but also we see increases in extensibility when wet. Now, this is because at a molecular level, water disrupts hydrogen bonds that occur between the molecular lattice, so that when water penetrates the fiber when exposed to wet air, it disrupts these connections between the amorphous structures, those alpha helices, beta turns, and this allows for greater stretchability because these structures can then unfold rather than potentially being damaged as they're bound together. And there are several examples of this in spider silk, lacewing silk, as I showed, as well as in glowworm silk, where, and I'll sort of show this later, but their nanocrystals are oriented in a sort of non-parallel direction to the fiber axis. And the idea is that they unfold similar to lacewing silk, where water actually penetrates the bonds between these beta sheets. And rather than stretching like spider silk, where the beta turns and alpha helices stretch, the crystals also unfold in both lacewing silk and glowworm silk, whereas in spider silk it does not. And this is a really unique behavior that has been hypothesized, but yet to be fully proven. Now, when wet, glowworm silk is also very sticky. This is really quite obvious, much in the same way like with the tensile properties. We can see here during our adhesion tests, when we've pressed down and we start pulling the fiber off of a force plate, we can see that as it extends over the course of several millimeters, we get a load value, a force value of resistance that increases as we stretch it. However, here we can see between the wet of these blue curves and the dry silks, not only, they really almost don't even stick at all to the force plate. And this is quite obvious from our statistical testing as well, that when pulled off of these platforms, there's almost no resistance at all, and that these silks just simply dry out and become unresponsive, whereas when they're wet, they are, fairly sticky over the course of several millimeters. And this is likely due to this drying effect, and if we look at these photos, it becomes quite apparent, where almost all of the weight due to water is reduced. We see they, in their natural condition, or after re-wetting, they look quite plump and full of water, but when dried, they start to crystallize and become far less obvious and shrunken and sullen. And you can see this is the final droplet that hangs at the end of the fiber that collects the most liquid. It's become shrunken, sort of crystallized with this root-like lattice. It's not a very pretty sight, and clearly these fibers suffer from that. And this demonstrates that these biomaterials that are produced by these glowworms are likely a representation of the first instance of functional dependence on caves. This may be an adaptation previously unknown to cave animals, and that, as I had said, there's many examples of adaptation to caves, like the longer life cycles and the elongated limbs, loss of eyesight, and pigmentation. It may also be that the biomaterials produced and the foraging capacities may also be adapted to cave environments, and this would be the first example of that at least I can point to. But these uses and exploitation of wet environments may have useful inspiration for design of synthetic adhesives, such as wet environments within the human body or other cave-like conditions, and possibly even underwater. And these sorts of exploitative characteristics of silk taking advantage of water is something that I will talk further in depth here in the next study, where we are surprised by the use of water rather by these insects and spiders. Because in large part, it's not exactly something that is expected by animals and by any adhesive to be able to use water in the case of using it to generate adhesion. So the first study that I presented with glowworms was more of a observation of the material and its behavior. In this study, we'll get to be a little more theoretical, but in a way that is we can talk about how these materials from a experimental and theoretical perspective perform in ways that we wouldn't expect. This study was recently accepted, so hopefully if you're interested again, you can go and take a look at what we did. If you're interested in understanding the concepts further. But from the most simplistic perspective, water is typically disruptive to adhesives. Think about a wet bandage that you may have encountered at some point in your life, or sweat, or rain, or water from some other source causes the bandage to become less sticky. This would be an adhesive failure, where the water molecules are actually binding to the adhesive molecules of the bandage or tape or adhesive itself. So this image simply shows the two substrates. So this might be the cloth or plastic of the bandage. The gluey adhesive would be this red, and then the substrate it's adhering to is this blue. And as the water binds to the adhesive, the adhesive is no longer able to bind to the substrate, causing failure and delamination or detachment. Water can also cause structural weaknesses in the glue itself, causing a cohesive failure, where the water molecules are better at binding to the actual molecules within the glue. And this is a big problem in epoxies and resins. As you can see here, these are big, deep cracks and imperfections formed by the water molecules being in place in improperly set and dried resin. It can be a major issue in many synthetic adhesives, and is something that materials engineers are really working hard to figure out a solution to. And so in order to overcome this issue, we can look to nature for some inspiration. And there's many examples, two of which we've already talked about with glowworms and spider silk, but also in mussel byssus and in cement produced by mollusks such as barnacles, where they're able to generate strong adhesion underwater. And in the case of our glowworms and spiders, can create strong adhesion in these super wet and damp environments in the caves. So understanding how these adhesives operate from these natural materials may help us inform development of synthetic materials. And as I had said before with cohesive failure, one way in which we can better understand this is to identify how animals exploit mechanical plasticization. Now, I'm not going to talk deeply about how animals use water to enhance the adhesive at the interface. I was more interested in the underlying materials and how water can help enhance that sort of structural compliance and extensibility. Because, as I had said previously, exposure to water disrupts these chemical bonds. This leads in synthetic materials to those cracks and imperfections that lead to quicker failure than if it's dry, whereas natural materials are robust to these changes. As I had said before, this causes an increase in extensibility, but also a lack of loss of strength. In this graph here, it's quite a colorful image, but if we look at going from purple all the way to red, these fibers are at a state where they were wetted originally and to various degrees. That's what these degrees mean. The higher degree is basically the more dry the fiber is, and therefore it's a little bit stiffer. But as it's wetted, it becomes stretchier. It does lose a little bit of strength, but this isn't catastrophic to the fiber itself and is still functional in many applications. So we wanted to know, does this make a difference in spider silks and in other types of silk? But the problem is measuring this and decoupling it from the effects of water the effects of water at the adhesive interface is quite difficult, mostly because, as you've seen before, these are either performed underwater or these materials have some kind of gluey, water-based adhesive that keeps the fiber hydrated. So we had to find a model that wasn't initially wetted, and criblate silk is the perfect model for this. These are silks that are similarly used in webs, and webs of all kinds, whether they're sheet webs or webs held by spiders in their arms. And they similarly function to entangle insect prey, much like those gluey threads, but they're composed of a different type of adhesive. They act on these dry adhesive fibrils, as I talked about a little bit before, that form these sort of puffs. And while it's difficult to see, these are actually hundreds to thousands of little nanofibrils that act like a glue, and they're supported primarily by these core fibers. So they operate as a composite with two different materials that operate as one. And spiders produce them through a combination of a specialized set of glands and a comb called the calamastrum on their hind legs. As they comb them out, that causes these puffs. And so instead of operating on a glue, they use van der Waal forces and hygroscopic forces. So these van der Waal forces are just near molecule forces where you have a difference in charge between the molecules, and hygroscopic forces are obviously sort of capillary forces, as they can use water. And however, there is a bit of debate as to the effects of water. In some cases, several studies have showed that there's a decrease in adhesion where the fibrils collapse, so that at dry conditions they seem to function rather well, but in when they're exposed to a fine mist, the water in the air causes them to bind together and collapse. However, other studies have shown that at higher humidities, they actually increase adhesion, and that adhesive force is increased. And so there's been some debate over the years, but that seems to have been somewhat settled due to possible differences in method, in that using this fine mist may cause large droplets of water and higher water saturation that causes these silks to bind together, rather than if we used evaporated air with lower water saturation, so lower content of liquid water, that may simply allow the interaction with water but not binding. So that may be what the differences in methods between these studies may have been what led to these, meaning that in wet conditions such as the humidity of caves and sort of subtropics and wet nighttime at dew point may increase adhesion, but in rainy days it may cause collapse and therefore non-functionalization of webs. A study that I had performed on 2 species of spider we had found that compliance, so the stiffness, dramatically decreases in silks from wet fibers rather than from dry fibers. So we were inspired to think, well, given that this is there is no previously the silks are not exposed to a wet glue or a underwater environment, we can control the plasticization of the fiber without artificially inducing it through those conditions that are pre-existing. So this is a perfect model for us to measure how mechanical plasticization impacts adhesion without coupling it with a previously confounding factor. So this brings us back to Hickmania. As I said, they inhabit these caves and these wet environments over the course of their entire life, which means that they're exposed to constant atmospheric water saturation, meaning that they're constantly exposed to wet conditions and they need to be able to catch prey in these wet conditions. So we thought, well, it's unlikely then that their silk should be collapsed or non-functional because they live in these conditions constantly. They use this cribbleate silk again in their webs to catch prey, and this is really quite important to them because because their metabolisms are slow and the food resources are low, they constantly need to be, they can't miss out on opportunities to catch prey. Their silk needs to perform well. So their silk is quite similar to what I showed. Here's a photo that we took, the spider in the web. You can see that the cribbleate silks are these dark, more bold looking fibers, and when they're fresh, they look a bluish color. This is a polarized image. Using light microscopy, you can see that there are these two fibers, and the nanofibril puff, which isn't in full focus, but it's this kind of pleated looking background that surrounds the fiber. And we showed that our methods do not change or cause the fiber to collapse. These are SEM and TEM images in dry conditions and wet conditions, where we show pretty obviously that these fibers don't collapse on themselves in either condition, which is great for our study because we can show definitively that it's unaffected primarily by our methods. So we wanted to test if the cribbleate silk is optimized for wet environments by looking to see if stickiness or adhesive work increases and if compliance of the underlying fibers increase. So once again, back to the caves, collecting the cribbleate fibers from the, and you can see the bluish hue there. We're collecting those pieces and then putting them onto the same machines and the methods similarly described as before, and running tensile tests. And when we expose them to wet and dry conditions, similar to how we talked about before. However, one thing is that the criblate silk fibrils were not manipulated, but they do not contribute until those axial fibers break. So meaning that their contribution is very low, so we can easily identify the actual performance of the underlying fiber. So from that, we were able to see a lot of interesting things. From we compared across body size because of, again, these spiders live in all together. So we compared these properties between two different treatments of dry and of wet across body size, similar to what we did previously with dragline silk. We found that diameter increases, again, not differently from what we'd expect from isometry, meaning that there's no particularly interesting changes in diameter of the silk compared to the size of the spider. And then we see changes in stress, meaning the strength. It actually gets slightly weaker as spiders age, and the extensibility increases across age, meaning that toughness is about the same, the overall energy. However, if we compare between our treatments, between wet and dry humidity conditions, we do see that our stiffness is very different, that in wet conditions, it becomes very compliant, much like a rubber band, as I had said, but in dry conditions, quite stiff. And that's quite easily identified here in our mechanical behavior graph, where we can see initially when you stretch it, the dry fibers are quite stiff, whereas when they're wet, they're very dry, very compliant. And this has big implications for our adhesive tests, as I'll show you in a second, because this initial area is under the same forces that adhesion occurs under, which would, obviously allow for those forces to potentially be dissipated. Now, our adhesion tests, again, very similar to what we did for glowworm testing. Samples were mounted above the force plate, lowered onto the plate at tension, raised at a fixed rate, where we measured force and extension up until fibers detached, also at these high and low humidities. So I have a quick video of those occurring. This is in dry conditions. As you can see, we pull up on the fiber and it detaches. Notice that the angle is relatively obtuse and that it doesn't deform very much. But in wet conditions, already at the same tension, it's much more compliant. You'll actually see here that the axial fibers are stretching, and we can see that this angle is more acute, though closer to a 90-degree. I'll go back just to, if you missed it, just to have another look at the difference between these pull-off behaviors. You can see again that these fibers, the axial fibers underneath, don't seem to deform very much, and that really they only bend slightly, and it seems to be those criblate fibrils, those blue-hued fibrils, that do most of the deformation. Whereas here, play the video once more, we can see that the underlying fibers and fibrils deform much more and actually do stretch while attached. And we can see it's much more dramatic. And that's quite clearly demonstrated in our data. We see that both across size and between treatments, there's a clear difference between how the silks perform. Greater adhesive force is generated, more extension of the overall fiber, leading to greater energy dissipation. Between tests, there was a slight difference. We tested these multiple times so that the first, second, and third generally showed a decrease in performance. However, that was generally not different in anything except adhesive force. But we can see that The dry silks didn't really perform very well and didn't really change much across size of spider that they were collected from, but the wet silks increased in performance across spider size and obviously can perform much better than their dry counterparts. When we run a correlation analysis and look at how these things perform, when looking at tensile properties and adhesive properties together, in our set of dry variables, we see no interesting trends, so we don't need to worry too much about it. In our wet conditions with performance of adhesive and tensile properties in wet conditions, we see that just adhesive extension and stiffness or compliance are pretty well correlated, which would indicate that when wet, both tensile properties of the axial fiber and extension of the thread itself are quite closely connected, which indicates that that compliance that I showed you is important, and it indicates some degree of transfer of those forces from the fibrils to the fiber, meaning that everything is working together. That the fibrils and the fiber are operating together, allowing for forces to be transferred rather than isolated to the fibrils. This would improve the overall performance of these threads so that when they're being used by spiders and webs, they can be better able to catch the prey that they're encountering. This would be facilitated by water and enhanced rather than disrupted, much like I had said before with the synthetic materials. This is something that doesn't generally happen. And this also allows for an interesting phenomenon where the forces aren't just located at the attachment site, but are actually being distributed throughout the entirety of the material. So generally speaking, in most cases, adhesion is only really important in the location where it's attached. But here, as we can see clearly at the right before the point of attachment, the forces that are being used, that are generated to resist detachment, are being distributed throughout the entirety, even the parts that aren't attached, which is something that is quite unique and helpful when you're trying to capture struggling prey. And we use the mathematical model. I'm not going to explain this because it's quite complicated, but it further illustrates and supports our conclusions here of mechanical plasticization, increasing mechanical work, as well as demonstrating the axial fibers' involvement and contribution to the overall composite material. So this may be somewhat of a novel mechanism, at least in this type of silk. May also be evidence of cave adaptation for spiders, not just glowworms, of their biomaterial to these kinds of environments. However, future testing needs to identify whether or not this is specific to this species or if it's a widespread phenomenon across cribbulate silks of other species, particularly those that do not inhabit webs or inhabit caves. And finally, this may be used to inspire future design of these of synthetics that are capable of exploiting water for its practical uses in mechanical plasticization that would limit the possibility of cohesive failures and disruption of internal molecular hierarchies, because this would then allow for enhancement of the material rather than degradation in these kinds of conditions. So finally, we've reached near the conclusion of our talk. I'm going to just simply try and spend a few brief moments convincing you or attempting to convince you of this new frontier in biomaterial research that we may have stumbled upon, and hopefully gain some inspiration and potential contact from people that may be interested in working on and collaborating on projects in the future. However, I don't want to give away all of my ideas because they're a bit premature and haven't yet been fully developed. So just to recap, we have 3 examples of how biomaterials research may be a frontier in caves, where we found that tensile properties of cave spider silk is increased across their lifespan. This would provide a unique model for future research to identify the mechanisms responsible for these observed shifts that may lead to improved development of fibers and other kinds of materials. And then the dependence of both glowworm silk and cave spider silk to these wet conditions to improve the performance of their silk. And in the case of spider cribbleate silk, how it is used to exploit those conditions to enable cribbleate mechanical plasticization that allows for improved performance. Other examples of biomaterials research in caves include further work that was done on glowworm silks, where this unique mechanism of unfolding of the nanocrystals in wet conditions may be something that is a design feature in future kinds of synthetics. And then there have been discoveries of the cave egg stalks of a species of spider known as Meta, in the genus Meta, that produce super stretchy silks due to the wet environments that can stretch, almost 800% of their original length, which is something that is quite incredible, as well as the discovery of or exploration in the cave has led to the discovery of unique spinning apparatuses in these Pilorempis flies that use these specialized structures on their legs to directly produce silk that they use to produce nuptial gifts that they offer to females for mating purposes. And so this is just a little snippet of the discoveries that could be further made that involve these biomaterials that can lead to discoveries that are important not just to zoologists and cave enthusiasts, but also to materials scientists and engineers that are looking to solve real world challenges of today. So there are several avenues that can be looked into. Again, following up the adhesion studies that we've already looked at, but figuring out what's going on at the interface with the interaction between water, the adhesive, and the underlying substrates of the fiber and whatever it's adhering to. Chemical composition is not very well known yet. It may lead to discovery of very unique biomolecules that we may try to synthesize for use. Further discovery and study of mechanics, better understanding the effects of water plasticization and other materials that are used in caves, and how spiders can respond to that variability because they're constantly exposed to these wet conditions. How do they remain functional, and how do they respond, and can they respond in different ways to these same conditions? And finally, as I alluded to at the very beginning, antimicrobial properties are quite well speculated on, particularly for these cave environment dwelling insects and arthropods, because as one example, Hickmania can produce egg sacs that will exist for up to 6 months before hatching, and those egg sacs then, once they're opened and fall away, are still not degraded immediately by microbial life. So there's likely some sort of antimicrobial properties that just remain undiscovered, but would be quite interesting for the use in human bodies, as well as other materials that might be produced that require those kinds of properties, such as disinfectants that are not necessarily harmful to good bacteria or other beneficial microbial life. So there are very many people that I'd like to thank for assistance and support during these studies, including my co-authors and collaborators. And for those of you that are interested in seeing more live action of the Tasmanian caves, I would highly recommend finding a way to see 16 Legs, which was led by and spearheaded by Dr. Neil Doran and Alistair Richardson. I was briefly featured in this movie for about 10 seconds, but it's really quite an incredible film that really does a good job talking about Tasmanian caves, the glowworms and spiders that live there, and all of the other amazing things about these caves. So with that, I will take questions. And for anyone further interested in looking up these studies or looking up any other research studies I've conducted, you can check me out on researchgate.com or Google Scholar. And I have be very willing to share directly with anybody that also is interested. So with that, I thank you for your attention and thank you for giving a chance to learn about spiders and glowworms and their silks.
Awesome. Well, thank you, Dakota. And I love that picture with the glasses. That's a studious spider there. That is cute. All right. Now let's see if anybody has questions. I've had just a couple come in. First one is, hang on, I texted them to you. First one was no cavemen. He said, no cavemen according to media. The other one was, what do you mean cavemen are not extinct?
Yeah, so, not so much to say the species of cavemen, so, Homo neanderthalis, but maybe the different human species that were known as cavemen, but my intention was more to mean the potential mindset of the cavemen may not be fully extinct. So sorry to make that unclear for those out there.
It just made me think. I have some friends in Gibraltar who found a lot of caves in Gibraltar, found the remains of walls and tools that were used by Neanderthals, which has kind of put the anthropology, the paleontology people up in arms for a little while until it proved that Neanderthals, that lived in caves actually had thought processes or doing some amazing things. And I wondered if you were talking along that line. Another question is, how in the world did you ever land on something like this for a thesis? What happened to where you ended up doing an entire thesis on this?
Yeah, that's a good question.
Your study, your passion.
Yeah, so this was something that I sort of stumbled onto. When I was an undergraduate student at the University of Akron, I had been working in the silk lab of Dr. Blackledge. And when I was in the course of my studies, I became interested in a study abroad program, and I spent a semester in Tasmania. That was my first trip there. And while I was there, I was interested in trying to conduct a research study. And I had done some, basic digging through literature to see what kind of interesting spiders might exist there that I might be able to provide some kind of justification for a study. And I pretty much immediately found the Higmania chalcarides, the Tasmanian cave spider, and thought, well, this looks like it'll be the best, the best model for a study. And I did some very basic rudimentary research there, and that's when I met Dr. Neil Doran, and I sort of spent time, going around to the caves and working there. And I thought, well, wow, no one's really doing anything here. There's these glowworms, there are these spiders. Nobody's really tried anything yet. This seems like a great system to work on. And so then during my time in Taiwan, we were able to get funding to go back a few times and conduct further research. And it's been great. This is a part of my thesis. There were some other studies too and other work that I've done as well, but this was some of my favorite work to do. And I'm, quite sad that I haven't been able to go back, but it's really, yeah, great question. And it really sort of stemmed out of my interest in doing research and sort of, learning about Tasmania. And actually, I was taking a class at the University of Tasmania, and Neil was a guest lecturer. And when I met him, I was like, wow, this is the guy he's already published on this species. He's sort of the go-to, and it all just kind of took off from there.
Okay, that's just amazing. All right, any more questions so that we don't hold Dakota too long, because he's on the other side of the world. It's afternoon right there where he's at. So do we have anybody else that has any more questions before we release him to go about during his day? Yeah, I don't see anything coming in. You can also reach him. There's his information on the screen, but you can also go to caves.org, and on the webinar section, you can email me and I'll forward everything to Dakota. Your invitation that came from GoToWebinar, same way. Put your information, your questions, your comments, your compliments, etc., and I will forward them to Dakota. But no other questions have come in. I'm going to let you go because it's already late out. It's already afternoon there. But thank you, Dakota. I really appreciate it, and especially all the time you spent for us trying to get the bugs worked out of this international webinar here. I greatly appreciate it. And I want to thank all those who attend. And remember, this will be posted on the NSS webinar section usually we get it up within 48 to 72 hours. So Dakota, thanks again, and good luck with the rest of your studies. And everybody, cave safely and be careful out there. Protect yourself from the virus. We'll talk to you soon. Have a good night.